Bismillahirrahmanirrahim. Assalamu alaikum. This is me Nazir Ahmad from Pakistan. Today our topic of discussion is drug clearance through metabolism. It includes phase 1 and phase 2 reactions and kinetics of metabolism. On my channel, please subscribe to my YouTube channel and press the bell icon button so that you may not miss any upcoming video. Okay, let's start. Drug clearance through metabolism. Once a drug enters the body, the process of elimination begins. The three major routes involved are hepatic metabolism, elimination in bile, and elimination in urine. Together, these elimination processes cause the plasma concentration of a drug to decrease exponentially. That is, at any given time, a constant fraction of the drug present is eliminated in a unit of time, as shown in figure. Plasma concentration versus time. Most drugs show an exponential decrease in concentration with time during the elimination phase. Most drugs are eliminated according to first order kinetics, although some such as aspirin in high doses are eliminated according to zero order or non-linear kinetics. Metabolism leads to products with increased polarity, which will allow the drug to be eliminated. Clearance estimates the amount of drug cleared from the body per unit of time. Plasma concentration versus time graph indicates extrapolation to time. Zero gives C0 the hypothetical drug concentration predicted if the distribution had been achieved instantly. The half-life, the time it takes to reduce the plasma drug concentration by half is equal to 0.69 volume of distribution divided by clearance. Total clearance is a composite estimate reflecting all mechanisms of drug elimination and is calculated as clearance is equal to 0.693 into Vd divided by half-life, where T1 by 2 is the drug's elimination half-life, Vd is the apparent volume of distribution, and 0.693 is the natural log constant. Drug half-life is often used as measure of drug clearance because for many drugs Vd is a constant. Kinetics of metabolism include first order kinetics, zero order kinetics. First order kinetics, the metabolic transformation of drugs is catalyzed by enzymes, and most of the reactions obey Michaelis-Menten kinetics equation. That is, V is equal to rate of drug metabolism is equal to V max into C, that is concentration of drug, divided by Km, that is Michaelis constant, plus drug concentration of drug. Uh, in most clinical situations, concentration of the drug C is much less than the Michaelis constant Km, and the Michaelis constant Michaelic Menten equation reduces to V is equal to rate of drug metabolism is equal to V max into C divided by Km. Here Km is the substrate concentration at which half of the maximum velocity is achieved, whereas Vmax is the maximum reaction velocity at which all enzymes become saturated with substrate. Zero order kinetics. With a few drugs such as aspirin, ethanol, and phenytoin, the doses are very large. Therefore, C concentration of drug is much greater than Km and the velocity equation becomes V is equal to rate of drug metabolism is equal to X into C divided by C is equal to V max. The enzyme is saturated by a high free drug concentration and the rate of metabolism remains constant over time. This is called zero-order kinetics, sometimes referred to clinically as nonlinear kinetics. A constant amount of drug is metabolized per unit of time, and the rate of elimination is constant and does not depend on the drug concentration. That is, the rate of drug metabolism and elimination is directly proportional to the concentration of free drug, and first order kinetics are observed as shown in figure. Here, with a few drugs such as aspirin, ethanol, and phenytoin, the doses are large. Therefore, the plasma drug concentration is much greater than Km and drug metabolism is zero order, that is constant and independent of the drug dose. With most drugs, the plasma drug concentration is less than Km and drug elimination is first order, that is proportional to the drug dose. This means that a constant fraction of drug is metabolized per unit of time, that is, with every half-life, the concentration reduces by 50%. First order kinetics is sometimes referred to clinically as linear kinetics. Reactions of drug metabolism. The kidney cannot efficiently eliminate lipophilic drugs that readily cross cell membranes and are reabsorbed in the distal convoluted tubules. Therefore, lipid-soluble must first be metabolized into more polar hydrophilic substances in the liver using two general sets of reactions called phase 1 and phase 2 as shown in the figure. Phase 1 reaction include oxidation, reduction and hydrolysis. Phase 2 reactions include conjugation products. Some drugs directly enter phase 2 metabolism. Conjugated drug is usually inactive. While in phase 1, following phase 1, the drug may be activated, unchanged, or most often inactivated. Phase 1 reactions. Phase 1 reactions convert lipophilic molecules into more polar molecules by introducing or unmasking a polar functional group such as hydroxyl or ammonia. Phase 1 metabolism may increase, decrease, or leave unaltered the drug's pharmacologic activity. Phase 1 reactions utilizing the P450 system. The phase 1 reactions most frequently involved in drug metabolism are catalyzed by the cytochrome P450 system, also called microsomal mixed function oxidases. Drug oxidized and become drug modified. Uh, the oxidation proceeds by the drug binding to the oxidized form of cytochrome P450, and then oxygen is introduced through a reductive step coupled to NADPH cytochrome P450 oxidoreductase. The P450 system is important for the metabolism of many endogenous compounds such as steroids, lipids, etc. For the biotransformation of exogenous substances known as xenobiotics, cytochrome P450, designated as CYP SIP, is a superfamily of heme-containing isozymes that are located in most cells but are primarily found in the liver and gastrointestinal tract. Nomenclature: the family name is indicated by SIP added to an Arabic number followed by a capital letter for the subfamily. For example, SIP3A. Another number is added to indicate the specific isozyme, as in SIP3A4. As shown in figure, there are many different types of enzymes SIP and their concentration present in body. Specificity: because there are many different genes that encode multiple enzymes, there are likewise many different P450 isoforms. These enzymes have the capacity to modify a large number of structurally diverse substrates. In addition, an individual drug may be a substrate for more than one isozyme. For isozymes, Four isozymes are responsible for the vast majority of P450 catalyzed reactions. They are SIP3A4 or 5, SIP2D6, SIP2C8 or 9, and SIP1A2. Considerable amounts of CYP3A4 are found in intestinal mucosa, accounting for the first pass metabolism of drugs such as chlorpromazine and clonazepam. Genetic variability: P450 enzymes exhibit considerable genetic variability among individuals and racial groups. Variations in P450 activity may alter a drug's efficacy and the risk of adverse events. CYP2D6, in particular, has been shown to exhibit genetic polymorphism. CYP2D6 mutations result in very low capacities to metabolize substrates. Some individuals, for example, obtain no benefit from the opioid analgesic codeine because they lack the CYP2D6 enzyme that O demethylates and activates the drug. The frequency of this polymorphism is in part racially determined, with a prevalence of 5 to 10% in European Caucasians as compared to less than 2% of Southeast Asians. Similar polymorphism have been characterized for the CYP2C subfamily of isozymes. Similar polymorphism have been characterized for CYP2C subfamily of isozyme. The addition of a US Food and Drug Administration black box warning to clopidogrel highlights the significance of this polymorphism. Patients who are poor CYP2C19 metabolizers have a higher incidence of cardiovascular events, for example, stroke or myocardial infarction, when taking clopidogrel. This agent is a prodrug and CYP2C19 activation is required to convert it to its active metabolite. Although CYP3A4 exhibits a greater than tenfold variability between individuals, no polymorphism have been identified so far. This P450 isozyme inducers. The CYP450 dependent enzymes are an important target for pharmacokinetic drug interactions. One such interaction is the induction of selected CYP isozymes. Xenobiotics chemicals not normally produced or expected to be present in the body may induce the activity of these enzymes by inducing the expression of the genes encoding the enzyme or by stabilizing the enzymes. Certain drugs, for example, phenobarbital, rifampin, and carbamazepine, are capable of increasing the synthesis of one or more CYP isozymes. This results in increased biotransformation of drugs and can lead to significant decreases in plasma concentrations of drugs metabolized by these CYP isozymes, as measured by area under the curve, a measure of drug exposure with concurrent loss of pharmacologic effect. For example, inducers, for example, vampine and anti-tuberculosis drug significantly decreases the plasma concentrations of human immunodeficiency virus HIV protease inhibitors, thereby diminishing their ability to suppress HIV virion maturation. Right side figure lists some of the more important inducers for representative CYP isozymes. This figure is include inducers that are phenobarbital, rifampin, and carbamazepine, dexamethasone, phenobarbital, phenytoin, rifampin. Consequences of increased drug metabolism include decreased plasma drug concentration, decreased drug activity if the metabolite is inactive, increased drug activity if the metabolite is active, and decreased therapeutic drug effect. Inhibition of CYP isozyme activity is an important source of drug interactions that lead to serious adverse events. The most common form of inhibition is through competition for the same isozyme. Some drugs, however, are capable of inhibiting reactions for which they are not substrates, for example, ketoconazole, leading to drug interactions. Numerous drugs have been shown to inhibit one or more of the CYP dependent biotransformation pathways of warfarin. For example, omeprazole is a potent inhibitor of three of the CYP isozymes responsible for warfarin metabolism. If the two drugs are taken together, plasma concentration of warfarin increase, which leads to greater inhibition of coagulation and risk of hemorrhage and other serious bleeding reactions. Note: the more important CYP inhibitors are erythromycin, ketoconazole, and ritonavir because they each inhibit several CYP isozymes. Cimetidine blocks the metabolism of thiophylline, clozapine, and warfarin. Natural substances may also inhibit drug metabolism. For instance, because grapefruit and its juice inhibits uh, CYP3A4, drugs such as nifedipine, clarithromycin, and simvastatin, which are metabolized by this system. persist in greater amounts in the systemic circulation, leading to higher blood levels and the potential to increase the drug's therapeutic and or toxic effects. Phase 1 reactions not involving the P450 system. These include amine oxidation, for example, oxidation of catecholamines or histamine, alcohol dehydrogenation, for example, ethanol oxidation, esterases, for example, metabolism of pravastatin in liver, and hydrolysis, for example, of procaine. Phase 2. This phase consists of conjugation reactions. If the metabolism from phase 1 metabolism is sufficiently polar, it can be excreted by the kidneys. However, many phase 1 metabolites are too lipophilic to be retained in the kidney tubules. A subsequent conjugation reaction with an exogenous substrate such as glucuronic acid, sulfuric acid, acetic acid, or an amino acid results in polar, usually more water soluble compounds that are most often therapeutically inactive. A notable exception is morphine 6-glucuronide, which is more potent than morphine. Glucuronidation is the most common and the most important conjugation reaction. Neonates are deficient in this conjugation system, making them particularly vulnerable to drugs such as chloramphenicol, which is inactivated by the addition of glucuronic acid, resulting in gray baby syndrome. Note drugs already possessing an possessing hydroxyl ammonia or carboxylic group may enter phase 2 directly and become conjugated without prior phase 1 metabolism. The highly polar drug conjugates may then be excreted by the kidney or in bile. Reversal of order of the phases. Not all drugs undergo phase 1 and phase 2 reactions in that order. For example, isoniazid is first acetylated, a phase 2 reaction, and then hydrolyzed to isonicotinic acid, a phase 2 reaction. Our next topic of discussion in presentation is drug clearance by the kidney. It includes glomerular filtration, proximal tubular secretion, distal tubular reabsorption, role of drug metabolism. Thank you for watching this presentation. If you have any question, please write in the comment section below.